Hi everyone, and welcome to today's webinar, Guidance for Cleaning, Disinfection and Sterilization of Reusable Medical Devices. I'm pleased to introduce today's speakers, Melissa Galle and Stefanie Roberfroid. Both Stefanie and Melissa have been working for several years in our microbiology lab and have a lot of experience in microbial testing, in vitro toxicology testing, and reprocessing validations. I now hand the microphone over to Melissa.
Thank you, Katrien, and thank you for the nice introduction. You probably noticed that the new medical device regulation was published by European authorities, which has, of course, some major changes as compared to previous regulatory documents that existed for medical device manufacturers. In this webinar, Stefanie and I will highlight the important points that are addressed to processing of reusable medical instruments. You will also get insight in the design of processing validation studies. As you probably already know, the current European Medical Device Directive and Directive on Active Implantable Medical Devices have been replaced by the new MDR, which was officially published on the 5th of May in 2017. There is no grandfathering for currently approved medical devices. So, the new MDR will be applied on all new and pre-existing devices from the 26th of May 2020 onwards. It is really key for the medical device manufacturer to know how to be compliant to this MDR and make sure to be prepared for that next year. Some of the key changes of the MDR that may have impact on processing of reusable devices in general are highlighted in this slide. First of all, the definition of a medical device has changed. It is expanded to also include devices without a medical intended purpose, for example, colored contact lenses and cosmetic implant devices and materials. Also be aware that for any device proportionate to the risk class and appropriate for the type of device, the manufacturer is expected to plan, establish, document, implement, maintain, and update a post-market surveillance system as part of the manufacturer's quality management system. So what the MDR wants is that the manufacturer plays an active role during the post-market phase. Of major importance is also the change in classification of medical devices, which is based on risk, contact, duration, and invasiveness. And finally, a recertification in accordance with the new requirements is mandatory for devices which are already certified under previous regulation. So to summarize, in a sense, the medical device manufacturer will be held responsible for the safety of the medical device during its entire life cycle, meaning from the first use until the end of life of the device. And therefore, it is key to make sure that you validate every step in the life cycle of a reusable device, meaning, first of all, the processing that has to be done prior to the first use, then the full processing cycle that will be performed to prepare the reusable devices for the next patient. This cycle includes all cleaning, disinfection, and sterilization procedures that have to be performed on the device. And finally, you are also responsible to determine the end of life of your device. In this webinar, we will focus on the full processing cycle of the reusable medical device. Here in this illustration, you can see an example of an average operation room. As a patient, imagine you are the patient. You want to be sure that your surgical procedure is performed under the best conditions. You definitely don't want to worry about the safety of devices which are marked with a red circle in this slide. For example, tweezers, scissors, even monitors. and therefore healthcare facilities need a processing cycle in place that typically can be summarized like this in the next slide. Preparation for decontamination of used instruments should already begin at the point of use. So with the point of use, I mean during the surgical procedure already or right after completion of the surgical procedure. Care at the point of use helps in preventing organic material, blood, and debris from drying on the surfaces and in lumens of used instruments. So, care at the point of use can improve the efficacy and effectiveness of cleaning. This is then followed by a proper cleaning procedure. This cleaning procedure may involve manual or automated cleaning steps and is performed as a precursor to a prescribed terminal disinfection or sterilization process. After cleaning, a disinfection procedure may be performed. This is mainly dependent on the type of the particular medical device. Cleaning and disinfection procedures are subsequently followed by a visual inspection and wrapping, after which the devices can be sterilized and then stored until next use. Every step of the diagram is pre-validated. It is really important that validated procedures are clearly translated to an instruction for use document to avoid any deviations when applied in healthcare facilities, because deviations from this may impact the safety of the device and eventually may compromise the safety of the patient, and this is what we do not want. It is simply not allowed to put unvalidated processing steps into the IFU. So, now we are going into more detail about the validation of the cleaning, disinfection, and sterilization procedures for reusable medical devices. Stefanie, can you please explain in more detail how cleaning procedures are typically validated for reusable medical devices?
Yes, of course, Melissa. It is first of all important to understand what is meant with the cleaning procedure, either a manual or an automated one. Cleaning procedures are designed to wash away any debris that comes from a patient and stay behind on the device or instrument during a surgical procedure. This can be, for example, tissue, blood, saliva, or sweat. When you are designing a cleaning procedure, it is very important to know the exact properties of your medical device itself really well, as they are key in the way a cleaning procedure will be performed later on. On this diagram, you can see the key steps in the validation of a cleaning procedure for reusable medical devices. And these include the design of the device itself, also the way a patient contact details when using during a surgical procedure, and also an assessment on how clean your device really is after we have performed the proposed cleaning procedure. The first question you always need to ask yourself is if you can provide devices that are already in a used state for the validation. A cleaning validation should always be performed on devices that are in a so-called used state. Devices that come straight from production may have some residues from the production process itself, and these can, for example, be lubricants or detergents that were used. And these may interfere with the analytical techniques that we are using during our validation to assess whether our cleaning procedure was successful. Furthermore, it is known that residues of detergents used during production can also interfere with your biocompatibility properties of the device. Also, any potential of soil that accumulates on the device will be assessed by performing simulated use testing. Therefore, Nelson Labs will always bring your devices in a used state prior to start a processing validation study in case we receive devices that are not in a used state yet. We frequently get questioned by medical device manufacturers whether they have to validate a full processing cycle for each and every device design that is produced. And I can comfort you that the answer is no. It's not always necessary. And I will explain this with a little example. Imagine a medical device manufacturer who is making drills like the ones that you can see on this picture. These drills can be very similar in design, with only having, for example, a different length and some small differences in the drill heads. And for this type of devices, it is allowed to group the device in different families based on a similar use of the device, the type of material they are made of, and also the size and challenging features that are present. And if you look at similar surgical procedures, the expected debris that will stay behind on the devices will be very similar. Furthermore, if material types are identical, they tend to hold debris in a similar way. Different material types are also known to hold on residues differently. And finally, based on the design of the device itself, a worst-case device for a family group can be identified, which we will then use during our validation of a processing procedure and will then represent the entire family. If you are designing new reusable devices, you should realize that some features that are incorporated in medical devices can be later on challenging to be cleaned. And some examples are given on this slide. For example, any textured surfaces can be very challenging to get cleaned. Debris from a surgical procedure, such as blood, for example, can stick on these devices and surfaces and dry into all small slits that are present. And the same problem you might have with, for example, braided cables. Also, any lumens and small pinholes may be difficult to reach during a cleaning procedure, so it makes them also very challenging to get cleaned properly. And sometimes multiple challenging features are even present on one device at once. For example, this monitor on the slide. We have here a handle with a structured surface, different types of buttons, and also multiple material types. So when designing new devices, it is always worth to keep these features into account, as they can cause problems in a later stage when processing procedures need to be validated. Also of great importance is the handling of reusable devices before and after cleaning, and especially when the devices can be disassembled. So it is very important to know how exactly and when in your reprocessing cycle devices need to be disassembled and reassembled again. Clear instructions, preferably with clear pictures and diagrams, should be provided and included in all processing validation protocols, as well as in the final instruction for use manual that will be available for the healthcare facility, so the end user of your device. Okay, we now have discussed in the couple of slides the design features of the medical device itself. And now we are going a little bit deeper into soiling procedures that are used during cleaning validation studies. Therefore, we will take always into account the type of patient contact to identify possible residues that may stay behind on your device after surgery, and we will mimic the real-life surgical procedure as much as possible to get really a representative soiling of your device. To make a reusable device dirty, we can use different ingredients, and you can see a list amongst others here on this slide. The type of debris that will be chosen depends on the clinically relevant marker that we will assess later on. For example, blood vessel clamps can come into contact with blood, so a blood source will of course be part of our testing soil. The relevant marker then to search for after cleaning will be hemoglobin. And we have, similar to blood, different ingredients that can represent any protein, carbohydrates, or lipids. Just to make this all a little bit less abstract, I have here a small case study. We got once a question from one of our sponsors to design a cleaning protocol for a collection barrel that was used during liposuction. And you can see an illustrative picture here on the slide, how it looks like after use. So the first question was then, what can we use as a test soil? And we tried some different recipes here in our laboratory, but nothing looked really similar to what you see on this picture. Eventually, we came up with a very clever soil, namely we combined butter and we spiked it with some blood to mimic the fat and the blood after patient contact. And after thoroughly mixing the test soil, we got actually a test soil that looked very similar and almost identical to what you can see on this picture. So this example clearly indicates that your composition of the test soil that you're going to use, together with the markers that you wish to test for, is determined by the intended use of the device. Our soiling procedures itself should mimic a real-life surgical procedure as much as possible. And you can see some examples here on how a real-life surgical procedure can be mimicked in a laboratory during a validation. Devices may be immersed in a test soil, either completely or partially, whichever is applicable during surgery. Furthermore, handling of devices with dirty gloves or devices that may get sprayed by blood or saliva can be mimicked in a laboratory during validation. And the experience of Nelson Labs allows to make sure that you're not soiling too little, neither too much, but represent a real-life situation as much as possible. After soiling, the reusable device in a representative scenario, a proposed cleaning procedure will be performed, and this cleaning procedure may be either based on a proposed instruction for use, a draft version that we get from our sponsor, or we can help you writing a protocol with our in-house experience. Cleaning of reusable devices can be performed in a manual, a mechanical, or an automated way, or even a combination of all these techniques. The technique that is chosen is highly dependent on the properties of the medical device itself. For example, manual cleaning steps may include soaking, brushing, or flushing. And flushing, for example, can be used to clean small pinholes or lumens that may be present on a particular device. Whenever, however, and keep that in mind, whenever the integrity of a medical device will not be compromised, it is obligated by guidances to have at least one automated cleaning cycle in a washer disinfector validated for that particular device. The choice of a suitable detergent is very important, and you should always keep in mind that the detergent that is chosen for a cleaning validation is compatible with your device and is available to the end user as well. When choosing a suitable detergent, always keep in mind that clinical conditions, as they will decide if you have to use an enzymatic or an alkaline detergent, for example, and what pH range should be used. Also, especially when manual cleaning steps are incorporated and involved in a cleaning procedure, your cleaning supplies that are used can be very important. These supplies should not only be compatible with the device. For example, it should be possible for the chosen supply to clean difficult to reach regions when used for that intention. But it also has to be easily available for a healthcare facility. Sometimes specific cleaning supplies can even be provided together with the device by the medical device manufacturer. When we are going to design a cleaning procedure at Nelson Labs, we will always tend to test in a worst case scenario. And that means that we, for example, are going to take the lowest allowed concentration of a cleaning agent. Or we take the shortest soaking time for the device. Flushing volumes will be chosen just under the minimum volume recommended in instruction for use of the manufacturer. And enzymatic detergents are used in certain temperature ranges in order to be active. So we choose at Nelson Labs to use, for example, the lowest allowed temperature in our cleaning validation process. By doing so and choosing for a worst case scenario, you take a human factor as well into account during your cleaning procedure, because in a healthcare facility later on, You can imagine that the shortest soaking time, for example, may be chosen by the operator that is performing your cleaning procedure just to save some time. After designing and performing a cleaning protocol, we want to make sure, of course, that the device is indeed sufficiently cleaned. And therefore, we have some analytical techniques in place to quantify possible remains of the test soil after performing the procedure. To be able, of course, to quantify any residues of your soil, the clean device will be extracted. Different extract methods can be used during the validation of a cleaning procedure to collect the remains of the soil in an appropriate extraction volume. During the extraction, different techniques may be applied depending on the device design and properties. For example, a flushing step can be applied for lumen devices. Manual and orbital shaking methods as well as sonication are very frequently used. And for very large devices or very specific device designs, for example, that have a touchpad or a hand palm scanner, something like that, a swapping technique of the contact surface can be applied.
And Stefanie, how clean is clean?
Well, that's a very nice question, Melissa. After extracts from the medical devices are collected, we will determine how clean they are, and therefore we are using the acceptance criteria that you can find in some of the guidelines here on the slide. They will tell you which amount of, for example, proteins is still allowed in the extract after the cleaning procedure. The amounts that are found for the relevant markers will always be compared to acceptance criteria that are applicable for the device itself and that were provided to us by our sponsor.
Stefanie, this information is actually very interesting. Let us have a look at an example.
Yes, I've prepared one example here. It was a case study about a blood vessel clamp. So let us just walk through it, how we are going to make the protocol for a cleaning validation study. First of all, we are going to choose the correct test soil. In this case, a blood vessel clamp will be in contact with blood. So a blood soil will be used because that is clinical relevant for this type of device. To soil the device, the tip was immersed in the blood soil. And furthermore, our analyst was using dirty cloths to take the clamp in its hand by the handle and started to open and close the device just to mimic a surgical procedure as much as possible. As this device, it can withstand a washing cycle in an automated washer disinfector, so we used actually an automated cleaning cycle to clean this device. After the cleaning procedure, the device was extracted by sonication and the extracts were tested for residues of following markers. So our sponsors chose for the markers protein and hemoglobin that were really relevant in this case study. We got the following acceptance criteria from our sponsor: 6.4 microgram per square centimeter for proteins, and we should not find more than 2.2 microgram per square centimeter for hemoglobin of the device in the extract. So to be able to have the test sensitive enough to quantify residues of proteins and hemoglobins below the provided acceptance criteria, we will have to calculate a maximum extraction volume that can be used based on the surface area of the device itself, the acceptance criteria that were provided by our sponsor, and the level of quantification of the analytical technique that we will use to measure remains of the particular marker in the extract. So just to summarize, what did we discuss so far? So we have validated a cleaning protocol to be used for a specific device and proved that any residue that remains on the device after performing that cleaning procedure are within acceptable levels. So we are now able to wash that device and wash away any remains of blood, mucus, saliva, or anything else. Okay, now the devices are cleaned, they are ready for the next step. So Melissa, can you maybe explain what will be taken into account during a disinfection procedure of a medical device?
Yes, of course, Stephanie. First of all, I want to highlight the significant difference between the process of cleaning and the process of disinfection. As Stephanie already explained before, cleaning is the process of removing contamination, such as residues of blood, other patient-derived debris, and germs from all surfaces, holes, and lumens of the device. So, a cleaning validation actually assesses the efficacy of a process designed to affect the physical removal of contamination from all surfaces. During a disinfection procedure, the microbial contamination on the device is diminished by killing microorganisms to an acceptable level and using an appropriate disinfectant. So a disinfection validation demonstrates the efficacy of a process designed to reduce microbial contamination present on the exposed surfaces of a reusable medical device through either chemical or thermal methods. So to summarize, cleaning is all about removing contamination, while disinfection is all about killing germs to an acceptable level. Why doing disinfection validations? Well, disinfection can be an alternative for sterilization methods if, for example, the device does not withstand common sterilization methods such as steam sterilization. Furthermore, it can be provided as an alternative to those healthcare facilities that do not have access to sterilization methods. As you already heard from Stefanie, for cleaning, you will need to choose an appropriate detergent. Well, for disinfection, you will need to select an appropriate disinfectant. And also here, it is important to choose products that are available to healthcare facilities and that do not compromise the integrity of the device when they are used in the intended concentration and contact time. Anyway, if a disinfection procedure is indicated in the instruction for use, it must be a validated method.
So Melissa, do I understand it correctly that the disinfection validation is very similar to a cleaning validation?
Yes, Stefanie, that is correct. But an important part is the neutralization study. So prior to the actual disinfection validation, a neutralization study is performed to demonstrate the absence of growth-inhibiting disinfectant in the extraction fluid and on the membrane, causing inhibition and false negative results. This is not what we want. If you have a look at the test setup of a disinfection validation, it is indeed similar to the setup of a cleaning validation. So first, the test items are artificially inoculated with a mixed suspension of test organisms. Then, the test items are disinfected using an appropriate disinfectant according to the supplier's recommendations and instructions. High-level disinfection is usually performed through soaking the device for a specified amount of time. Glutaraldehyde solutions or hydrogen peroxide-based disinfectants may be used for this. Intermediate and low-level disinfections are usually performed by wiping or spraying the devices, occasionally by soaking. For this isopropyl alcohol, quaternary ammonium compounds, KV-side wipes, for example, or spray may be used. After disinfection, the test items are extracted for bioload reduction evaluation. And how do we do such a bioload reduction evaluation? Well, the bioburden of the test items is determined by immersing and shaking the test items in a sterile extraction fluid. The extraction fluid is then split into equal parts. Each part is filtered over a membrane, and each membrane is then aseptically transferred to an appropriate agar plate. Following a specific incubation period, the colonies on the plate are counted, and then the log reduction of the inoculum can be calculated. As you can clearly see here in this slide, there are different levels of disinfection, namely high level, intermediate level, and low level disinfection. We can help you determine what level of disinfection is appropriate for your device based on its use and where it fits in the Spalding classification. This classification makes a difference between critical, semi-critical, and non-critical devices. First of all, we have the non-critical devices. These are devices that are either non-patient contacting devices or devices that come into direct contact with the intact skin of the patient. Use of these devices carry relatively little risk of transmitting an infection directly to patients, but still can be a source of microbial transmission. A monitor, for example. A monitor has no direct patient contact, but can be categorized as a non-critical device. So for a monitor, cleaning followed by a low-level disinfection procedure may be sufficient. Then we have semi-critical devices. These traditionally come into contact with mucous membranes or damaged skin of the patient. Devices belonging to this category may require intermediate level or even high level disinfection. Flexible endoscopes and urinary catheters are examples. Critical devices are devices that are introduced directly into the human body during a surgical procedure, so either into the bloodstream or sterile tissue. Devices belonging to this category may require high-level disinfection or even sterilization between patients. Scalpels, transfer forceps, and cardiac catheters are examples of critical devices.
So Melissa, and how do you assess that after a disinfection validation, your devices are really well disinfected? Are there also acceptance criteria like it's for cleaning validation?
Yes, indeed, Stefanie. So for each level of disinfection, there is one or more acceptance criteria. So a 6-log10 reduction of 4 vegetative bacteria separately is required for a low-level disinfection. For an intermediate level disinfection validation, there is an additional requirement. A 3-log10 reduction of mycobacterium needs to be achieved to demonstrate a reduced risk on tuberculosis. A high-level disinfection validation is successful if a 6-log10 reduction of mycobacterium is demonstrated together with an A0 value of 600 for critical devices. After the disinfection validation, we have not only washed away any debris like blood, but now also microbes are killed to an acceptable level.
So for those devices that only need the cleaning and disinfection step and no further sterilization step, these are now ready to be used to the next patient. They are considered to be safe. Is that correct?
No, actually not. So there might be a potential problem. So a potential problem that may occur with processing procedures, where we use detergents and disinfectants. Well, residues of these chemicals may stay behind on the devices. So the biocompatibility of the processed device cannot be guaranteed anymore. Therefore, Nelson Labs will perform a cytotoxicity assay to monitor biosafety of the devices that have been processed. The big advantage of a cytotoxicity test to monitor biocompatibility during processing validations is that the test is not only cheap, but also fast and sensitive. After a completed cleaning and disinfection procedure, the device is extracted in MEM complete medium, which is compatible with the cytotoxicity test system. So then our cells are exposed to the extract of the device and subsequently, after appropriate incubation, of course, scored for their viability. This can be done with qualitative or quantitative methods. Anyway, a device should pass the cytotoxicity assay after processing. In case a failure is detected, Additional rinsing steps may need to be included in the cleaning or disinfection procedure to get rid of any remains of chemicals. Many reusable devices are sterilized after cleaning and disinfection, or even immediately after cleaning, depending on the type of device. The purpose of sterilization is similar to that of disinfection to some extent. Both techniques have the purpose to lower the amount of microbes on the devices. Well, both techniques actually do this by killing. Where for disinfection, for example, a 6-log-10 reduction of vegetative bacteria needs to be validated, sterilization has a much stricter acceptance criterion. A product is considered sterile when a sterility assurance level of at least 10 to the -6 can be shown. Pre-vacuum steam sterilization is validated to a sterility assurance level of at least 10 to the -6, meaning that there is less than a 1 in 1 million possibility of a contaminating organism surviving the treatment. There are many sterilization processes available, but the sterilization process predominantly used in healthcare facilities is steam sterilization. Here in this slide, you can see why steam sterilization is so popular in healthcare facilities. The process is non-toxic and dependable. In addition, steam sterilization is not only relatively cheap, the process is also characterized by a rapid culling action, and this is what we want. During the design of a reusable medical device intended to be repeatedly sterilized and reused, manufacturers should direct their attention to compatibility of materials with the sterilization process, but also product configuration and product functionality. Selected materials should be compatible with the specific conditions or potential effects of the process. So for steam sterilization, the materials should be resistant to heat, distortion, warpage, and corrosion. The materials should not only be able to withstand high temperatures, of course, but also changes in pressure. The design of the device should allow full steam penetration and should not hinder air removal or moisture elimination. The functionality of the device before and after steam sterilization should be exactly the same. That's also very important. It's also important to consider the challenges that long lumens, areas of high material density, mated surfaces, for example, but also assembled pieces and dead space create for the sterilization process. We always advise to limit blind holes, but also dead-ended lumens as much as possible. If you have a tray, it is important to reduce the total mass. Ideally, the mass is also distributed throughout the tray. Ensure a proper steam flow throughout the tray. You can use holders, that's not a problem, but make sure that these parts have limited contact with the device and do not block steam. Also make sure that the devices in the tray are not stacked on each other. This slide gives you a high-level overview of our approach when it comes to validating a steam sterilization cycle. As I already told before, pre-vacuum steam sterilization is validated to a sterility assurance level of at least 10 to the -6. And the preferred method to do so is the biological indicator overkill method. Why? Well, I will explain this in the next slide. But first, after inspection of the product, the worst case locations for air removal and steam penetration are determined as a first step. Then, depending on the feature to be inoculated, the inoculation method is selected. Next, a biological indicator carrier material is fixated on each inoculation site of the product under a laminar airflow. An appropriate type of packaging is then selected to wrap the product properly. The packaged products are placed over the cold spot of the steam sterilizer. That's typically over the drain closest to the door. After the half cycle time and all other relevant parameters are set, the sterilization process is initiated. Following the completion of the half cycle, the product is removed from the autoclave and placed under a laminar airflow to cool down for the safety of the technician. Then finally, all biological carrier materials or the inoculated products themselves are aseptically submerged in growth medium and then incubated at appropriate conditions. After incubation, the growth medium vessels are then inspected for the presence of microbial growth. In the next slides, I will go into more detail for each of these steps. The overkill method is preferred because the bioburden is not always the same. The bioburden of the device is different for various patients and environments. Depending on the feature to be inoculated, the most appropriate inoculation method is selected. We can choose from wires, sutures, paper strips, or suspensions. Each carrier material typically contains at least 10 to the power of 6 CFU of Geobacillus stearothermophilus. Carrier materials are then placed in the most difficult to sterilize areas, and examples of such areas are, for example, lumens, channels, narrow or occluded cavities, Here you can see an example of how we would determine the worst case locations for air removal and steam penetration. So in this slide, you see a large tray filled with many devices. Well, for this tray, we have selected 7 sites distributed across the entire tray. These sites were inoculated with sutures. And why did we choose sutures? Well, these are ideal to introduce in very small lumens. After inoculation, the product is actually ready to get packaged. There are different types of packaging, but not only the type of packaging, but also the size of the packaging is important. We do not want a packaging that is too tight, because then there is a higher risk for ruptures. First of all, we have the pouches. Pouches are typically used for lightweight medical devices. Next, sterilization wrap. Sterilization wrap is available in multiple sizes and different strength levels. Therefore, it is also suitable for heavier products. As a general rule, the higher the number, the stronger the wrap. A product can be wrapped in 1 or multiple layers using an envelope or square folding technique. The wrap must allow steam penetration, adequate air removal, and drying. And finally, we also have the rigid containers that come in many shapes and sizes and materials. They typically have perforated openings for ease of sterilization.
Melissa, can you please also elaborate a little bit about the differences between the US market and the EU market as it comes to steam sterilization cycles?
Yes, of course, Stephanie. So once the product is packaged, the actual sterilization process can begin. But there is indeed a difference between the EU market and the US market. Here in this slide, you can see the parameters for pre-vacuum steam sterilization cycles that are typically used in healthcare facilities in the US. These are also listed in the TIR 12 guidance document version of 2010. In this slide, you see the parameters for the EU market. If a device will be marketed in the EU, for example, a temperature of 121 degrees Celsius or 134 degrees Celsius is common. For more information about European dynamic air removal steam sterilization cycles, you can consult ISO 17665-2 version of 2009. After sterilization at the half cycle parameters and cooling down of the product, all biological carrier materials, or of course the inoculated products themselves, are aseptically submerged in growth medium under a laminar airflow. We would typically use trypticase soy broth for this. Then, the growth medium vessels are typically incubated at a temperature between 55 and 60 degrees Celsius for at least 7 days. This range is ideal for growth of Geobacillus stearothermophilus. After incubation, the growth medium vessels are all inspected for the presence of microbial growth. In the picture on the right bottom of this slide, you can clearly see the difference between growth in the left tube and no growth in the right tube. For a successful steam sterilization validation, we definitely want to have growth medium vessels without turbidity, as it is the case in the right tube. An important part of the validation of a steam sterilization cycle is the verification of the dry time at the end of the cycle. Dry time testing ensures that after the steam sterilization cycle, the packaging and devices are not wet. Moisture may compromise the sterile properties of the packaged device. Wet conditions always stimulate microbial growth. The dry time at the end of the steam sterilization cycle is verified by comparing the weight of the pouch, or the wrap of course, after sterilization to the weight before sterilization. After sterilization at full cycle parameters, the pouch or the wrap and the device are also visually inspected for the presence of any moisture. There are 3 acceptance criteria that need to be fulfilled in order to have a successful dry time verification. So first, to start with, all validation cycles must be within specification. The dry time is verified in triplicate just to demonstrate reproducibility. Secondly, the dry time will be considered validated when no visible moisture is observed on any surfaces of the packaging or its contents. And finally, the calculated weight gain percentage should be less than 3% for each of a minimum of 3 dry time cycles. In addition, it is possible to monitor the temperature within the sterilization chamber and the product during a sterilization cycle. Therefore, thermocouples are placed inside the product and sterilization chamber. During a full sterilization cycle, the temperature inside the product and the chamber are then monitored. Just to give you an example, if you look at the temperature profiles on this slide, you can clearly see that there is a difference in the ability of different materials to heat up to a desired temperature. Metal parts heat up very quickly compared to, for example, silicon parts. Stefanie, can you now please wrap up and conclude this webinar by summarizing the key points and some takeaways?
Yes, we are going to conclude with some key take-home messages. Be aware that as a medical device manufacturer, according to the new MDR, you will be the one that will be held responsible for the entire life cycle of a medical device. So this means from the first use of the device over the entire life cycle, including all processing and until the end of life of your device. Be also aware that each step that is written in the instruction for use manual provided with your reusable device regarding to processing of the device has to be validated in advance. A validation study should always reflect a worst-case situation. Device design and properties determine the procedures that are used for cleaning, for disinfection and sterilization, and should be taken into account when designing processing studies. And be aware that whenever you are using chemicals during processing of a device, these may cause alterations in the biocompatibility of your device. Therefore, cytotoxicity testing is used as a screening marker to assess any potential problems with biocompatibility, especially for those devices that do come into direct contact with the patient. So when you're following this advice, you will be able to have your processing procedures validated and in place, making sure that your devices are safe to be used for the next patient. Thank you for watching this webinar. In case you should have any questions, please do not hesitate to contact us by email or visit our website to find more information about Nelson Labs and the services that we can provide.